Friends, today we are going to study about internal structure of monocotyledon leaf. The typical example of monocotyledon leaf which we are going to visualize is the maize. The other examples of monocotyledon leaves to visualize, they are like sugarcane, maize, jawar, rice, wheat, bamboo. These are the examples of the monocotyledonous leaves which are very long and broad. So, of these photosynthetic larger long organs, we have to study how internally they are composed, how internally the photosynthetic tissue is there, what structure the photosynthetic tissue is shows, and what are the different types of vascular bundles present in the transverse section. Now, beside this, we have to observe what type of mechanical tissue is there inside the leaves of these monocots. Does the vascular bundle they shows any kavach bundle sheath around it? So all these things we have to observe. Now, the leaf as a whole, when we observe it, the maize leaf or jawar leaf or sugarcane leaf, it shows midrib or a midvein, very long, broad and long. And along the side of the midvein or midrib, there are the side veins running parallel up to the length of the leaf. So whenever we cut this transverse section of such a leaf, these veins thus running in the lamina of the leaf, blade of the leaf, they appear as vascular bundles in the transverse section. The veins they appear as the vascular bundles in the transverse section of the leaf. So this we have to observe there in the transverse section of the monocot leaf. Now over here, this is the transverse section of the monocot leaf, grass or maize we can call. This transverse section shows two skins, the upper skin, this is the upper skin, this is the lower skin. This is why the leaf is called bifacial. Upper surface is there, lower surface is there. This is a bifacial dorsiventral organ of the plant body. This is a photosynthetic organ which is very broad, thin, and very long photosynthetic green part of the plant body. We call it as bifacial dorsiventral organ of the plant body, which is photosynthetic part of the plant body. So we have to study the internal structure of it. Now, whenever we take the transverse section of such a bifacial organ, it shows upper epidermis. This is the upper epidermis. This is the upper epidermis, upper skin of the leaf, upper surface. This is the skin of the leaf, upper epidermis. This epidermis is composed of single layer of the cells which are compactly arranged without intercellular spaces except at the places where there is a stoma, where there is a respiratory stomata is there, singular stoma, plural stomata. So except at the region of the pores or the stomatal openings, the epidermis is continuous protective layer. Epidermis is a continuous protective layer. So, as usual, the function of the epidermis is the protection. The function of the epidermis is the protection, the entry of the pathogenic organisms, and to protect the body from the mechanical destruction. This epidermis acts as the skin of the leaf, protecting the internal delicate tissues. Now, to perform this function of protection nicely, this particular epidermis is deposited with the cuticle. This particular epidermis is deposited with the cuticle. So this is the cuticle covering the epidermis. The epidermal cells are covered with the extra coating on it. We called it as a cuticle. Now this cuticle is made up of different substances in different plants. Over here, The silicon salts are there in the form of cuticle over here, there in the monocotyledons. So this is about the upper epidermis. Now this particular epidermis, thus it is made up of barrel-shaped or dumbbell-shaped cells, barrel-shaped cells, cylindrical cells, which are compactly arranged and are of usual rectangular size. Now this particular region shows at certain places a balloon-like very larger cells. Suppose over here, here. There is a bunch of the cells, 2, 3, 4. They appear just like a fan, just like a bulging balloon. These are called as motor cells, or these are also called as bulliform cells, balloon-like, very larger. These motor cells or bulliform cells, thus present in the epidermis. These are with very larger cavities inside, which stores the water when it is available. Now these motor cells or bulliform cells thus present in the epidermis, these have the ability to fold the leaf and or to roll the leaf when the soil conditions are placid, when there is a scarcity of moisture, water in the soil. The plant body should be protected. So in such situation, these motor cells just present in the upper epidermis, what they do, they roll the maize leaf, they roll the jawar leaf, and or the lamina is folded, and due to the folding or rolling of the leaf, the leaf gets protected from the burning radiations of the sun. So to avoid the burning of the leaf, and due to the less moisture in the soil, placidity of the soil, these motor cells thus present in the upper epidermis help in protection or ruling of the leaf. And when the field or the soil is supplied with the water, these motor cells becomes turgid in the outline, and when the turgidity occurs there in the plant body, the leaf again it unfolds, the leaf opens, unfolds. So this is a sort of rolling and unrolling, folding and unfolding of the very long broad lamina of the leaf. It can be brought about by due to the activities of these motor cells, rolling and unrolling or folding and unfolding of the leaf, depending upon the availability of water moisture in the soil. When the soil becomes dry, the leaf folds to protect it from the solar radiations which are burning, which causes the burning effect. And with the availability of water in the soil and with the turgidity of water in the plant body, the leaf again reopens, unfolds, unrolls. So this is called as a rolling and unrolling of the leaves, brought it about by the motor cells, which is the characteristic property of the motor cells. Now below the motor cells there is a mesophyll tissue. This green is a mesophyll tissue. This is a photosynthetic tissue. This mesophyll tissue is of a spongy parenchyma type. The cells are loosely fleshed, just like a sponge. There are intercellular spaces just like the sponge, where the intercellular spaces are numerous. Over here, this spongy mesophyll photosynthetic tissue shows the porous nature, loose arrangement of the green photosynthetic chlorophyll containing cells, chlorenchyma cells. We call it as a mesophyll type of the tissue. Now this mesophyll type of the tissue, just below the upper epidermis, slightly it is compactly placed, and the cells are somewhat radially elongated. In fact, but all this is a spongy parenchyma type of photosynthetic tissue. Though the cells are radially elongated below the epidermis, this is a spongy parenchyma type of photosynthetic or mesophyll type of the tissue containing the chlorophyll pigments in it. Now, in addition to the mesophyll, the internal structure, if the leaf, if we observe, it shows the vascular bundles in it. Suppose over here, this is a vascular bundle of the main vein. This is the vascular bundle of the main vein, and in addition to the vascular bundle of the main vein, this is the main vascular bundle. This is the vascular bundle of the side vein running parallel. This is the vascular bundle of the side vein running parallel along the length of the leaf. Now the single vascular bundle, if you observe, it shows its elements like a xylem. This cavity is the xylem. This is the xylem. This is the xylem elements over here. Metaxylem we can call. The smaller one is called as protoxylem. Now, in addition to the xylem, there is a phloem over here. This is the phloem over here. Now, the phloem is toward the lower side and the xylem is toward the upper side. Xylem is above and the phloem is below. So, xylem and phloem, it is conjoint and it is collaterally placed. The vascular bundle is conjoint and collateral. There is no cambium in it. Therefore, the vascular bundles are said to be closed type of vascular bundles. Now, such vascular bundles, xylem and phloem, shows a bundle sheath. This red color is the bundle sheath. This is the bundle sheath. This is the kavach or protective sheath protecting the xylem and phloem, which is a conducting tissue present in the form of vascular bundle. Now, this particular bundle sheath over here, this is the bundle sheath. This particular bundle sheath shows above over here and toward the lower side. There are flanking, broadening patches of. This is the broadening patch of the mechanical tissue toward the upper epidermis. The same just toward the lower epidermis. This is the broadening patch of mechanical sclerenchyma tissue. This is the mechanical sclerenchyma tissue, or we can call it as an extension of the bundle sheath toward the upper epidermis and toward the lower epidermis. This is a sclerenchyma present. This is a sclerenchyma type of tissue present, flanking, broadening toward the upper epidermis and lower epidermis. This sclerenchyma tissue is a very harder mechanical tissue present above the vascular bundle and below the vascular bundle toward the lower epidermis and toward the upper epidermis. Now, just as the patches over here and here, there are these mechanical patches of sclerenchyma. They may appear over here, and these are the vascular bundles of the side veins running. So this is the same structure where the side veins also show the same feature. This vascular bundle and this vascular bundle, they are the same, but this is of a main vein and this is of a lateral vein. And just like this sclerenchyma over here, this particular vascular bundle may show over here the sclerenchyma patch, over here the sclerenchyma patch is thin. So this is about conjoint, collateral and closed type of vascular bundle of the monocot leaf, showing protective flanking patches above and below. Internally, the leaf also shows the lowermost lower region, lower skin of the body, lower skin of the leaf. We called it as lower epidermis. Lower epidermis. Epi means outermost. Dermis means a skin. Outermost lower skin. This is the outermost lower skin. This outermost lower skin is a protective made up of one layer of the cells which is compactly arranged. Barrel-shaped cells are there which are compactly arranged, and it also shows at certain places is the pores opening stomata we can call. Aperture is present in the lower epidermis. Just as in upper epidermis there is a pore or opening for respiration and transpiration, the lower epidermis also shows the pores or stomata for transpiration. So over here we can say that both in the upper epidermis and the lower epidermis, the stomata are present. The stomata, they show the stomatal cavity over here. This is a respiratory cavity or substomatal chamber we can call. This is called as substomatal chamber. This is called as substomatal chamber or respiratory cavity. Now, so this is all about the internal structure of the monocotyledon leaf, which is the bifacial dorsiventral organ of the leaf. Now, to identify the internal section of the leaf as monocot, we can identify it with the presence of such a characteristic type of bulliform or motor cells. When they are seen in the epidermis, we can call this as the section of the monocot leaf. So presence of the motor cells or bulliform cells in the epidermis indicate that this is the monocot leaf. Now beside this feature, to be a monocot leaf, the monocot leaf shows in both the epidermis, the upper epidermis shows stomata over here, the lower epidermis, it shows the stomata over here. So in both the skins, upper skin and lower skin, there is a stomata. There is a stomata. Now beside this, to be a monocot leaf, this particular leaf shows a characteristic type of mesophyll tissue which is of spongy parenchyma type. This is a spongy, loosely fleshed intercellular species, sponge-like photosynthetic tissue which is called as spongy parenchyma type of mesophyll or photosynthetic type of the tissue. So this is all about the internal structure of monocot leaf. Typical example which we have visualized over here is the maize. Typical example which we have visualized is the maize. Now with this I conclude over here the internal organization of the monocot leaf. If you have any doubts about the internal structure composition of the monocot leaf, you can ask me the questions on the group. So with this I stop over here. Dhanyavad.